learned about the different methods of contraceptions. In last class we have learned about the contraceptive devices, and we have learned about the permanent or surgical methods of contraceptions. So today we discuss about MTP, or medical termination of pregnancy or induced abortion, and the different technologies which has been discovered to help infertility. Yes, so first of all, what is MTP, or medical termination of pregnancy? So we should call this MTP or induced abortion. Write it down. MTP means medical termination of pregnancy. It is also called induced abortion. So what is MTP? So definition is, MTP is the intentional or voluntary termination of pregnancy before the fetus becomes viable. That means when the developing fetus has certain types of genetic disorders or any type of trait, if it possesses to the mother, then a married couple can go for MTP. But to go for MTP, alright, this calculated, but to go for MTP, the fetus must not be viable. Viable means self-living. Yeah. So MTP or induced abortion means intentional abortion or induced abortion. Clear? Before the fetus becomes self-living or viable. Now, what is the safe time for MTP? So the safe time to undergo MTP or induced abortion is during the first trimester of pregnancy. First trimester means up to 12 weeks. Up to 12 weeks of pregnancy or during the first trimester, MTP can be done. It is safe to be done. But after that, during second trimester, when the fetus becomes part of the maternal tissue, then MTP cannot be done. It will be a risk for the mother also. So the safe time for MTP to be done during the first trimester of pregnancy is up to 12 weeks of pregnancy. Clear? So safe time is first trimester or up to 12 weeks of pregnancy. Now why is MTP done? Why is induced abortion done? So there are different reasons for the MTP to be done. So what is the first reason? Number one, casual unprotected sexual intercourse. Alright, if the sexual intercourse is done without using any physical barrier contraceptive, then it will lead to unwanted pregnancy. So in that case, one can go for MTP. Second one, failure of contraceptive devices. If the contraceptive devices will fail to function, then it will also lead to unwanted pregnancy. Next one, pregnancy due to rape. And fourth one is the most important one, where there will be a serious threat to the fetus, mother, or both. So when the pregnancy will lead to a serious threat to both the fetus and the mother, and in that case one can go for MTP or induced abortion. Clear? So both of what is MTP, the full form of MTP, and remember the safe time for MTP to be done. Clear? And why is MTP done? Now mainly in India, right, or in most of the countries, let's say, there are different types of laws which has been imposed to mainly deal with the MTP. But mainly in India, MTP was done by the technique of amniocentesis, which we have learned in the previous classes. Try to recall, where the amniotic fluids will be taken, the chromosomal patterns will be seen, and depending on that, the sex of the fetus can be known. Clear? Though it was used to identify the genetic disorder of the fetus, but at the same time, amniocentesis was also being useful in knowing the sex of the developing fetus. And if the fetus is to be a female, then one is to go for the female fetus size. Clear? So nowadays there are different type of laws which have been enacted to mainly control the medical termination of pregnancy, right? And prenatal sex determination has also been banned. It's also illegal. It's illegal to find out the sex of the developing fetus. Okay? So remember what is MTP, the sex time for MTP, and the reasons why MTP is done. Clear? Now next one. What is infertility and sterility? So infertility means the inability to conceive or produce children in spite of unprotected sexual intercourse. If a married couple will not be able to conceive a child even after the unprotected sexual intercourse, then that particular inability to not conceive a child is known as infertility. Alright, but what is sterility? It is the complete inability to produce children. The complete inability to produce a children is called sterility. But what is infertility? Is mainly the inability, not complete, is the inability. This can be dealt with the different types of devices, different types of methods which we'll discuss afterwards. How to deal with infertility, alright? How to help those married couples to mainly conceive a child. That's called ART. We'll discuss after this one. So infertility is also inability, but it can be tackled, it can be dealt to produce children or to conceive children in spite of unprotected sexual intercourse. But sterility means the complete inability to produce the children. So how can the infertile couples can conceive a child? Clear? So before that, let's talk about different reasons for infertility in male and female. Why are they not able to conceive a child? Okay, so there are different regions of infertility. So mainly in males, what can be the reasons? Mainly in men, the reasons for infertility can be due to the low sperm count. Try to recall that in a single ejaculation, nearly 3 to 4 ml of semen will be released, and each semen contains around 200 millions of sperms. Clear? Now, if the number of sperm counts will be less, clear, it will also be a problem. Or if the motility of these sperms will be very, very poor, it will also a problem for fertilization. If these sperms will not be functional, there will be also a problem for conception. Clear? So there are different reasons for infertility in case of men. So let's look over here. A man is infertile if the semen has low sperm count, number one, low sperm count, or abnormal sperm structure, or the poor sperm motility. So this can be the reasons for infertility in case of male. Now in females, it can be due to irregular ovulation, or no ovulation, or defect in the genital tract, like the impaired motility of the fallopian tube, defective uterine endometrium, try to recall the endometrium part of the uterus, defects in the cervix, defective vaginal growth, or the deficiency of sex hormone. So there are different reasons for infertility in case of male and in case of females. Clear? Next one. Terms related to male infertility. So let's go to different terms or disorders in case of males. First one, oligospermia. Clear? Go through it. Continue. Oligospermia is the condition of low sperm count. So low sperm count means oligospermia. Near absence of live sperms. It's called azoospermia. Low sperm motility. It means spermatozoospermia. Defect in sperm structure, teratospermia. Cryptorchidism means failure of testes to descend into the scrotum. And last one, hyperthermia means higher temperature in the scrotal sac. So these are the different terms which has been linked to male infertility. Clear? Oligospermia, azoospermia, stenospermia, teratospermia, cryptorchidism, and hyperthermia. So kindly go through these terms. Next one, ART. Now what does ART means? ART means assisted reproduction technologies. Now these technologies are mainly used to help those infertile couples to conceive a child. Alright? So those who are infertile, those who are not able to conceive a child even after undergoing unprotected sexual intercourse. Right? These particular technologies, ART, can be used for those couples to mainly conceive a child. So ART includes a number of techniques which assist infertile couples to solve their infertility problems. So ART is mainly a technology, right, which can be used, which can be very helpful for the infertile couples to mainly conceive a child. Now, next one. What are the different technologies or the techniques in ART? First one, IVF-ET. IVF means in vitro fertilization. IVF means in vitro fertilization. ET means embryo transfer. So in vitro fertilization, embryo transfer. Second technique, IUT. IUT means intrauterine transfer. ZIFT, ZIFT means zygote intrafallopian transfer. Next technique, GIFT, GIFT. GIFT means gamete intrafallopian transfer. AI means artificial insemination. Next technique, IUI. IUI means intrauterine insemination. And ICSI, the last technique under ART, ICSI means intracytoplasmic injection. So these are the different techniques that comes under ART, which can be used for the treatment of infertile couples. Okay, so we'll go through it one by one. So first of all, we'll discuss about IVF-ET. So IVF-ET means in vitro fertilization embryo transfer. So I've written this particular slide from the textbook only. So kindly go through it. Okay. So let's figure out what is IVF-ET or in vitro fertilization embryo transfer means. It's also called test tube baby. In this case, in IVF, the fusion of gametes, the sperm and the egg, occurs outside the female body. Put it down. In IVF, the fusion of sperm and the ova occurs outside the female body in the laboratory conditions. That's what is called in vitro fertilization. Clear? And there is called test tube baby. So we'll go through details over here. This is the technique of fertilization of an ovum with sperm carried outside the body of female in a glass container. In vitro fertilization. So try to remember it. So in IVF, the fusion of sperm and the ova occurs outside the female body in the glass container in the laboratory conditions. Clear? It is called in vitro fertilization. So let's go with the procedure of IVF. The procedures involve the following steps. So first step, so we'll go through it one by one. So drugs are administered to prospective mother to induce or development of several ova, which is called superovulation. That means in point number 1, the female will be administered with a drug that contains the female hormone that will help in superovulation. Now what is superovulation? Generally, in a normal menstrual cycle, right? During ovulation, that means on the 14th day of the menstrual cycle, one ova will be released from the ovule. Clear? But in superovulation, multiple ova will be released, will be formed, more than one, or you can say more than two. So more than two ova will be released. Alright, so that's what it's called superovulation. So first point, drugs will be administered to the female, which will lead to superovulation, where there will be formation of multiple ova. Number 2, just before actual ovulation, the mature oocytes are retrieved. That means before ovulation, the mature oocytes will be retrieved. They will be collected. Clear? Second, third one, the retrieved mature oocytes are placed in culture medium containing nutrients. So those ova which has been collected before ovulation, which are mature, now they are placed in the culture medium containing nutrients. The culture of oocyte is kept in incubator for about 4 to 6 hours. So what we are doing? We are administering the drug to the female body, which will lead to superovulation. And just before ovulation, the mature oocytes will be collected from the female's body and will be cultured. Alright, and will be cultured in the culture plate containing the nutrient medium. Then, after that, the culture of oocyte is done in the incubator for how many hours? For 4 to 6 hours. Now, after that, semen that contains the sperms. So, semen to be used for insemination. What is insemination? Is the release of sperm into the female genital tract that will lead to fertilization. Now, in this case also we will require semen. Clear? So, semen to be used for insemination, to be used for fertilization, is purified, concentrated by hose and by swim technique. And nearly about 50,000 to 1 lakh fully functional sperms are taken. Clear? So, we are going for the selection of the sperm. So, around 50,000 to 1 lakh fully functional sperms are taken. Now the ova is ready, sperms have been taken. Okay, now we will go with the fusion. So now the sperm and the ova will be fused. So purified semen is poured over the mature retrieved oocytes. Now just we have to add the sperm over the collected oocytes. Eggs are examined under the microscope for about 12 to 24 hours after insemination. Since fertilization occurs outside the female body in a culture medium, it is called in vitro fertilization. The fertilized eggs show 2 pronuclei in a second polar body. So what we have done? So we have taken around 50,000 to 1 lakh fully functional sperm. Now that sperm will be used to fertilize the oocyte, which is present in the culture medium. So fusion is done. Now over here, the fusion of sperm and the oocyte does not occur inside the female body, but is occurring outside the female body. In which medium? In a culture medium, which is called in vitro fertilization. The eggs will be examined continuously under the microscope, and we'll see that the egg which is fertilized will show 2 pronuclei in a second polar body. Now fertilization is completed. Yeah. Next one. The fertilized eggs are selected and incubated for about 48 to 72 hours in an incubator. During this period, fertilized egg or eggs undergo cleavage and reach 48 cell stage. Now, what does 48 cell stage mean? Try to recall. Zygote undergoes mitotic cell division, 2 cell stage, 2 to 4, 4 to 8, 8 to 16. Remember, morula, blastula, right? A cell with 16-30 stage is called morula. Morula changes into blastula. Okay, so those fertilized eggs will be selected and will give an incubator. Now that fertilized eggs undergo cleavage and will reach the 48 cell stage. After that, the fertilized egg is stimulated to develop in vitro up to 32 cell stage. That means the egg will keep on dividing mitotically from 2 to 4, 4 to 8, 8 to 16, 16 to 32. Alright, will keep on dividing. This embryo is implanted on the endometrium of the uterus. That means when the egg will reach 32 cell stage, then it will be implanted into the endometrium of the uterus. So at 32 cell stage, the egg will be implanted into the endometrium of the uterus. The female has now conceived and is pregnant. Further development of the child continues in the womb till it is born. Further baby is called test tube baby. So what we have done? Just briefly. First of all, a mother's body will be administered with the drops that mainly contain the female hormone, leading to superovulation. Clear? Superovulation means where multiple ova will be released. Then sperm is to be taken. Semen is to be taken. It has to be concentrated by wash up and steam up technique. Then around 50,000 to 1 lakh fully functional sperms will be taken. Then that selected sperms will be utilized to fertilize the egg. So what we do? Before ovulation, the secondary oocytes, the mature oocytes will be taken from the female body, will be placed in the cultured medium, and that selected sperms will be used to fertilize the ova in the cultured medium. Now this fertilization occurs outside the female body in a cultured medium, so it is called in vitro fertilization. Next thing, now the egg will be fertilized, it will be examined under the microscope. It will keep on dividing metrically from 1 to 2, 2 to 4, 4 to 8 to 16, and those fertilized egg will be kept in incubator. Then this egg at the 30 cell stage will be implanted into the endometrium of the uterus. Now that females will conceive a child and become pregnant, and such child, such baby which are born by this technique is called test tube baby. And in case if the mother is not able to conceive a child, in that case we need the help of a surrogate mother. Clear? So this is very technique of IVF. Clear? So next slide. Many have learned that egg at the 32 cell stage are implanted into the uterus of the female. But depending on the stage of embryo transfer, at what particular stage the embryo will be implanted or will be transferred to the female's body. So generally, we have got two types of IVF. One is called IUT, intrauterine transfer. It depending on the stage at which the embryo is transferred to the female body. So first one is IUT, intrauterine transfer, and second one is ZIFT, zygote intrafallopian transfer. So what is the difference between IUT and ZIFT? So in IUT, the transfer of embryo is done in the uterus at 32 blastomere stage. That means when the egg cell is at 32 cell stage, in that case, it is implanted or transferred into the uterus. So when the egg cell at 32 blastomere stage is implanted, is transferred into the uterus, then it is called IUT, intrauterine transfer, done at 32 blastomere stage. But if the zygote or the embryo at 8 blastomere stage, 8 blastomere stage is transferred in the fallopian tube, then it is called zygote intrafallopian transfer. That means in this case, in ZIFT, the zygote or the embryo is implanted in the fallopian tube at the 8 blastomere stage. So in IUT, the embryo is transferred to the uterus, in the uterus at 32 blastomere stage, and in ZIFT, the zygote or the embryo is transferred to the fallopian tube at 8 blastomere stage. And this final technique, ZIFT, was developed by Devery in 1986. Next one. GIFT, gift, gamete intrafallopian transfer. Okay, let's see what technique means. In this procedure, the washed sperm and the two retrieved ova, that means the collected ova, are transferred into the fallopian tube with the help of a laparoscope, mainly a device. Okay, it's mainly used to examine the internal organs. Therefore, fertilization occurs in the fallopian tube. This technique is used in those cases where husband has very low sperm count. That means in GIFT, what we are doing, we are taking the gamete, the male gamete and the female gamete, and the gametes are being transferred into the fallopian tube. Yeah, so we will collect the male gamete, we will collect the female gamete, and those gametes will be transferred into the fallopian tube, and fertilization occurs in the fallopian tube. And this is mainly done in those cases where the number of sperm count is very, very low. So in this case, we need a sperm donor. Yeah, and those sperms can be mainly introduced into the fallopian tube along with the ova, which will ensure fertilization. Next technique, AI, artificial insemination or intrauterine insemination. Now before that, what is insemination? Insemination is mainly a technique, it's mainly a process where the sperm or the semen is released into the female genital tract. Yeah, but over here, we are going to talk about artificial insemination. So artificial insemination is a technique to treat infertility by placing sperm into the vagina or the cervix when woman is ovulating using a thin flexible catheter. That means artificial insemination is mainly done by using the semen or the collected sperm. Yeah, it's mainly placed into the female genital tract when the female is ovulating. At the time of ovulation, the semen is mainly released into the female genital tract. When you remember, the collected semen or collected sperm. The sperm then travel into the fallopian tube where they can fertilize the egg. AI is carried out with sperm from the male partner or a donor and can be combined with superovulation. Donor sperms are used if male partner is sterile, has extremely low sperm count, or carries risks of some genetic disease. So what is AI? AI is mainly a technique where the collected sperm is introduced into the female genital tract at the time of ovulation and mainly performed if the number of sperm count in the male is very, very low, or if the sperm mobility is very, very poor, or if there is abnormal structure of sperm. So in that case, we require a male donor or the sperm donor. Yeah? Okay. Now what is IUT? So artificial insemination is also called intrauterine insemination, IUI, in which sperms are introduced directly into the uterus. That means the collected sperm or the semen can also be introduced directly into the uterus. In couples with unexplained infertility, IUI increases chances of pregnancy many more. But in what conditions AI is recommended? Okay, we have just gone through the definition of AI, but in what conditions AI is recommended? Okay, so let's get out the conditions. So AI is performed when the sperm count is very, very low, or when there is absence of sperm in the semen. Number 2, when the movement of the sperm is very, very low. Number 3, when the man shows retrograde ejaculation. That means when the semen and the sperm are released in the urinary bladder. Next one, if the male partner shows ejaculation dysfunctions, so one can go for AI. In case of female, the cervical condition is hostile for the survival or inward movement of sperm. Last one, presence of any cervical scar tissue in case of female, which will hinder the sperm ability to enter the uterus. So these are the conditions in which AI or IUI can be done or is recommended. So in today's class, we have discussed about MTP, medical termination of pregnancy or induced abortion. We have learned the same period for induced abortion or MTP. Then we also seen the conditions where MTP can be done. Then we have gone through the different regions of male fertility, infertility, and the female infertility, and the different terms which has been related to the male infertility. Then we have learned about the ART, assisted reproduction technologies, which will help in reproductions. Yeah, so under ART, we have learned about IVF, ET, then we have learned about GIFT, okay, GIFT gift, then we have learned about artificial insemination also. So we will continue in the next